I think what this means is that this cell, which of course underlies everything we do, it's sort of beautiful.
I would like to ask you if you could briefly explain in very simple terms what your lab is focusing on.
Sure.
And what are the short-term and long-term implications on human health?
Okay, so I'm a neuroscientist, and we're very interested in the main cell of the brain, the neuron, and in particular how it manages to function in terms of its proteins. So unlike other cells in the body that are round, the neuron has its cell body, but it has this amazing set of processes, the dendrites and the axons, which are most of the volume of the cell. And so what that means is that it's a challenge for this brain cell to make and degrade the proteins that are mostly out in the periphery. And we've been studying for decades now how that happens, and we've figured out that it happens by putting the machines to make the proteins and turn them over way out instead of having them centralized. And you said for human health?
Yeah, let's say short term and way longer terms.
Okay, so I think what this means is that this cell, which of course underlies everything we do, it's sort of beautiful. It's made this morphology so it can massively connect with all of the other cells, which is what we need for neuronal networks, but it's also a vulnerability at the same time. So the beauty of it and its interconnectedness gives rise to susceptibility to disease because of this enormous challenge of getting the proteins out. So what we are trying to understand is very important because the process of supplying proteins and turning them over is one of the things that we know goes awry in many neurodegenerative diseases.
Probably your main discovery showed that proteins can be synthesized locally at dendrites when they are needed. And this is a crucial step for synaptic plasticity. Now, my question is, how did you arrive at that discovery?
We were doing experiments in brain slices, and we were recording electrical activity. And in the hippocampus, we were recording very far away from the cell body. So they're all lined up in a row, and we were hundreds of microns away. And we had established in that brain slice plasticity, so an increase in synaptic transmission. And we did an experiment to see if newly synthesized proteins were required, with the expectation that if we blocked protein synthesis, it would take a long time to see the effect of that inhibition because we were so far away from the cell bodies, which at the time was the presumed site of protein synthesis. But instead, what we found is that there was an immediate requirement for protein synthesis, which told us that the protein synthesis source needed to be right next to where we were recording, so local, as you say.
I see. And what do you think has changed since that discovery in the entire brain research community, I would say?
Well, those of us that are interested in the cell biology of the neuron, I think it has really changed the way people think about the processes and where these machines are and what can be regulated, because as you suggested at the time, the dominant view was everything was happening in the cell body, and so it was all about trafficking. It was all about moving things from the cell body out to the processes. So this really changed the view and said, actually what's moved is the messenger RNA, but the proteins are made locally, and later we and others also showed that they're turned over, so degraded locally as well.
I see. Thank you. Next one is, I would say the starting point is one of the sentences that is more used and I would say also abused in research, that is seeing is believing. And you actually develop some techniques that allow us to see actually this protein synthesis locally. So my question is if you can briefly and simply explain us how they work.
Yes.
And what they have allowed us to discover further.
So early on we made reporter fluorescent constructs where we used information that we knew about how mRNAs were getting localized to target those reporters. with the idea being that the mRNA goes out, and then when the protein is made, you see a fluorescent spot. So you could instantaneously, more or less, tell where the protein was made. But a larger body of work really built on the idea that if you could label the building blocks of protein synthesis, you could then see the proteins. And this had been exploited with radioactive amino acids, but of course, nobody wants to work with radioactivity anymore. So we were really fortunate when my lab was at Caltech that there was a chemist there who was working on amino acids that had been functionalized with chemical groups, azides and alkynes. And these two chemical groups can interact in a special kind of chemistry called click chemistry. And so what we did was to take those amino acids, in this case something that's like a methionine but has an azide group on it, and we know that during protein synthesis that can get incorporated instead of methionine. So you have now in the polypeptide chain that's being built a chemical handle, if you will, that can be recognized by the other group. And then, depending on what you add, you can visualize them again with a fluorescent molecule, or you can pull out. So then you can, if you want to know what those proteins are, you now have a way of pulling them out of the tissue and analyzing them with methods like mass spectrometry.
What are the, in your opinion, the major still open questions in your field, and where do you think the research should go in the next coming years?
So I think we and others have been really good at identifying all of the molecular players. You know, we know all of the messenger RNAs that are there. We know which ones are translated. We even know the sequence elements that are presumably responsible for their localization. But we don't really have a good handle on what I would call the logic. So how is it that certain mRNAs are transported and delivered to certain sites? When we look at the nucleotide sequence, there are a few consensus sequences that emerge that can account for some small fraction of the localized mRNAs, but not nearly enough. So that's one issue, which is, what is this logic? Is it combinatorial? How are the RNAs being recognized by RNA binding proteins? And what's the code for localization? Another important issue is related, which is how are mRNAs selected for translation? So, intrinsic to all of the beauty of this idea is that different patterns of synaptic activity and the presence or absence of different neuromodulators have the ability to select certain mRNAs to be translated. And we still don't understand how that selection process works.